To continue with our second half of fatty acid metabolism, we're going to talk about the synthesis of ketone bodies and then the synthesis of fatty acids themselves. So we're going to begin with acetyl-CoA. So acetyl-CoA is what we get out of degrading fatty acids, but in this case we're going to use the acetyl-CoA as our building blocks to make ketone bodies or to make other fatty acids. So the first thing, let's talk about the ketone bodies. So we're going to take acetyl-CoA with a lack of oxaloacetate. We can't enter the citric acid cycle, so we need to convert it into a more stable form. Remember, acetyl-CoA has that thioester linkage, which is not incredibly stable. So we need to turn it into a more stable form. One way to do that is to remove the CoA from it, but the acetate molecule in itself is not a good molecule for storage. We'd like to get some carbons connected together. So we're going to combine two, eventually a third, and then release the third, or the first, acetyl-CoA and make acetoacetate, and finally hydroxybutyrate, and these are our ketone bodies. We'd like to prevent forming acetone as a byproduct of this mechanism, and we'll see how we do that by reducing the molecule. So when would this happen? Well, we normally get lots of ketone bodies formation during starvation because there's a lack of glucose intake. But that can be mimicked by two other conditions. So untreated diabetics would also have a lack of glucose in the cells. We're not talking about in the blood here, but we're talking about in the cell. So in the cell, from its point of view, there is a lack of glucose in diabetics because the issue is not that there's not any glucose, it's all in the blood and can't get into the cells. Also in alcoholics, while not necessarily a lack of glucose, but an excess of ethanol. Ethanol being converted to acetyl groups, acetyl-CoA. And so in the cell again, there's a disproportionate amount of acetyl-CoA compared to oxaloacetate. So looking at the figure at the bottom, we're going to take two acetyl-CoAs, one in red, one in blue here. And the one in red is going to have its methyl group deprotonated and attack the carbonyl of the blue one. Okay? Leaving the CoA as an excellent leaving group. I connect the the 2 carbons to the other 2 carbons, and now I have a 4-carbon aceto-acetyl-CoA. Okay, in the second step, we introduce a third green acetyl-CoA, which again is deprotonated on its methyl group and attacks the very same blue carbonyl. In this case, however, we don't reform the carbonyl. It stays as an oxyanion, and a water molecule will protonate it to make it a hydroxy group. Okay, at this point, it is a tertiary alcohol because it is branched. Right? At the other end of the molecule, where the green CoA is attached, the remaining part of that water molecule, instead of the proton that was donated, we have a hydroxy anion, will attack the carbonyl there, releasing the CoA. And so in the figure here, you see the water molecule is shown in the black font, where the proton on the hydroxy making the alcohol group, and the other OH, which becomes a deprotonated because it's a carboxylic acid, at the very end on the green. At this point, we can go one of two ways. Later, we'll see how we can take this 3-hydroxy 3-methyl-glutaryl-CoA, which is HMG-CoA, and make squalene and ultimately cholesterol when we're talking about cholesterol synthesis. But at this point, we're going to lose the beginning acetyl-CoA to make acetoacetate. So all 3 CoAs that have been introduced here have left, either in the form of CoA or still attached to an acetyl group, and I have acetoacetate. One pathway here shown to the bottom is a spontaneous non-enzymatic decarboxylation, which would result in CO2 and acetone. So this organic solvent would not be a good idea to make in abundance, as it would disrupt our membranes. So instead, the cell will take the acetoacetate, consume an NADH to reduce the acetoacetate to 3-hydroxybutyrate. And that is a highly soluble form of our acetyl-CoA stored. Right? It's a storage form. So this is one of our ketone bodies that the cell will use. Okay, so we use it as fuel. And mainly I said this is in the form of when you don't have an input of glucose or during starvation. So it is mimicked in cases of alcoholics or in cases of untreated diabetics, but we're going to talk about starvation here. So looking at the chart at the bottom, after the first few hours to a day or two of starvation, the amount of glucose starts to drop. This is glucose in the blood. So the concentration of glucose in the blood will tend to drop, but it doesn't drop drastically. There's a smooth transition to it leveling out. Well, we're not getting any new glucose intake, but we can convert lots of things to glucose. Number one is going to be our stores of glycogen. Right? So the liver has lots of glycogen storage, and it can be mobilized into glucose. Okay, once that's been extinguished, our cells are going to need another source of glucose. So a lot of our non-essential proteins will be degraded into amino acids, and a lot of those amino acids can be converted to glucose. Only two of them cannot. We covered that in another lecture. Right, so we convert this degradation of protein into glucose, and mostly coming from our long muscles, right? Because we don't need those in days of starvation. We'd rather survive. So by the time we get to day 4, day 5, day 6, our glucose is leveled off because we have a steady stream of protein degradation to make more glucose. We also have a lot of fats, right? And we know fats cannot be converted into sugars in animals. So you cannot convert acetyl-CoA back into pyruvate. That reaction is completely irreversible. So we can't go back. We can't get through there. So we're going to convert the fatty acids we have stored, our fats storage, into acetyl-CoA. But since we can't make that into glucose, we'll turn it into these ketone bodies we just described. And you see post day 3, the amount of ketone bodies in the blood has spiked. And the chart on the right illustrates the source of fuel for certain cells and tissues. So fuel used by the brain on your 3rd day of starvation is still mostly glucose, about 100 grams of glucose a day, and only about 50 grams of ketone bodies. And by day 40 of starvation, assuming you have a water source, right, because you will not make it 40 days without water, but without food, you possibly could survive 40 days. Your brain is consuming mostly ketone bodies as opposed to glucose at that point. Okay, so on to making fatty acids. Instead of making ketone bodies, we'll convert these acetyl-CoAs into fatty acids for storage. Now, this is not during starvation. This is when you're well fed, but for some reason you need to make a lot more fatty acids, possibly for preparation for hibernation, for cells dividing, needing new fatty acids, or just a storage form of it as energy. Okay, so we're going to follow along as we make acetyl-CoA into a brand new long-chain fatty acid. The liver, unfortunately, will do this in times of, say, alcoholics or in times of type 2 diabetics. In this case, the cells again are lacking in glucose. Not in the blood, but the cells themselves aren't receiving enough glucose for the amount of acetyl-CoA they have. So what the liver tries to do is compensate by sending out more glucose. At some point, the liver will say, we need to make fatty acids. So the fatty acids made by the liver, because of the excess acetyl-CoA, trying to store it as a means of getting rid of the excess, will make so much fatty acid or fatty tissue, the liver becomes what we call fatty liver. That's totally reversible at this point if the diet changes. But if it gets too bad, the cells that are dying from the excess fats will become fibrotic, and that's called fibrosis. And that'll leave scar tissue if that's reversed in time. It's still recoverable. If this continues unchecked, you'll eventually get what's called cirrhosis. And the cirrhosis is cells that's completely damaged beyond repair, and eventually the liver cells and this liver will die. So let's start from the beginning in making fatty acids. We're going to start with acetyl-CoA. And acetyl-CoA, mainly from degradation of other things, is going to be found in the mitochondria. Well, mitochondria was where we did fatty acid degradation. However, we'd like to compartmentalize this and do fatty acid synthesis in a different area, not in the same compartment, so we don't get the two processes confused or competing. So we're going to move this acetyl-CoA into the cytoplasm to do fatty acid synthesis there. So we need to move acetyl-CoA across the inner and outer mitochondrial membranes. Well, the CoA part of acetyl-CoA is not amenable to crossing a membrane easily, so we need some kind of shuttle to get it across, but not in the form it exists currently. So much like other shuttles, we're going to convert our acetyl-CoA into a more recognizable form by one of our toll booths. In this case, we're going to combine the acetyl-CoA with oxaloacetate to make citrate. And if you remember, that's step 1 of the citric acid cycle. We're not going to continue the citric acid cycle here. We're just making citrate. And citrate can be exported to the cytoplasm in one of our toll booths that allows you to export one thing while importing another. We export citrate if I import a pyruvate. So that's what it does. On the outside, the citrate can be converted back to oxaloacetate and acetyl-CoA, which is the reverse of that first step. It generally does not go backwards in the mitochondria, but in the cytoplasm, there's an abundance of citrate coming out and very little oxaloacetate. So we can convert it back. We convert the oxaloacetate back to malate, again, just like we did before when we were talking about the gluconeogenesis pathway. It was the other direction. So here we're going to convert it to malate. And malate can easily be converted back to pyruvate. And then we have our pyruvate to import back into the mitochondria. Yes, we do lose a CO2 in that process, but we do gain an NADPH from the reduction, or from the oxidation of malate. At this point, pyruvate back in the mitochondria can be converted to oxaloacetate. This is step 1 of gluconeogenesis, and we've seen this before. We just carboxylate it back to oxaloacetate. Right, so we've got our acetyl-CoA moved from the mitochondria into the cytoplasm, where technically we only moved the acetyl part of it, so our CoA pools have remained separate. It makes it easier to do. And in the next step, we need to take our acetyl-CoA and start building the fatty acid. It's only 2 carbons long at this point, so we need to make it longer. At the moment, it's not so unstable. It does have that thioester linkage, but the 2 carbons are rather stable. It's just an acetate. So in the first step of dedicated fatty acid synthesis, we're going to take the 2-carbon acetyl-CoA and carboxylate it to malonyl-CoA. And we're going to add a CO2. That's not an easy reaction to do. It's going to require an input of energy in the form of ATP. And we've seen this before where we use ATP to activate a molecule. Then the molecule gets attached to something else at the expense of the ADP or AMP, depending on the reaction, leaving group. Okay, we did this with activating amino acids before they went on the tRNAs. We did this with glucose before we put it on the growing glycogen chain and so forth. So we'll do this again here. We're going to activate a carboxyl group with ATP. We're going to make a carboxyphosphate. Then the phosphate group is an excellent leaving group to attach the carboxyl group to the acetyl group, making a 3-carbon malonyl-CoA. Okay, this is the committed step in fatty acid synthesis. Once you do this, this is destined to become a fatty acid. Okay. So that enzyme that does that is called acetyl-CoA carboxylase, and it's one of the classic ping-pong or 2-site or double displacement mechanisms. Okay, so it happens where you have the first molecule coming in. We're going to call it Q in this picture. Right? I'm missing an O at the top of the figure, but we're going to call it Q in this picture. And in this Cleveland diagram, which represents the reaction progress, not necessarily time, the enzyme binds to molecule Q. Q does some sort of chemistry with the enzyme, possibly leaving a piece of itself behind. And then the product O, which is what's missing from the top of that arrow at the top of the screen, there should be an O leaving. At this point, the first product has been released, and the enzyme has been modified somehow. A piece of the original molecule has been left behind. Okay, in our case, we're going to see that's a carboxyl group. But the enzyme is modified. It can't undergo that chemistry again. In the 1-site ping-pong mechanism, a second substrate would come in and immediately take that piece away. In this case, we have a 2-site where this thing is transferred either to a second active site or a second enzyme, and it's the second reaction that occurs in a different location. So we're going to move it over to the second site, and in that site, the second substrate comes in, recognized by, or not recognized, but shown by P here in the diagram. So P comes in, binds to the enzyme, and it does the second half reaction, or the second chemistry, where it picks up that leftover piece. In our case, it's a carboxyl group, and then leaves as product R. Right? So I like these letters because you can follow the little slanted piece as it goes from Q onto the enzyme, leaving O left over. Then P comes along, picks up that little piece, and becomes R, regenerating the original enzyme so it can do this 2-site thing again. Okay, so apologize at the top. It should be an O on that leaving arrow. What does Q, O, P, and R represent here? Well, the enzyme is our acetyl-CoA carboxylase, so I'm representing it by the letter E, and it means either the biotin carboxylase, which is the site 1 of this enzyme, or the carboxyl transferase, site 2, plus the carrier of the biotin, which is a biotin carboxyl carrier protein, the BCCP acronym, and it's got a biotin attached to it. biotin's where our carboxyl group is going to be held. And so in the first half reaction, we have our biotin carboxyl carrier protein with its biotin group attached, and that's associating with the enzyme biotin carboxylase. In bacteria, these are all separate components, but in eukaryotes, this is all one big polypeptide chain. So you can think of it either way, but the steps are the same. So in the first step, our first substrate, Q, binds. Well, Q here is actually represented by a couple molecules. It's a magnesium-bound ATP, and it's a bicarbonate moiety. So both of those things are represented by Q. Okay, so they come together. The enzyme causes the carboxyl group to get phosphorylated by the ATP, making a carboxyphosphate. And then the carboxyphosphate decarboxylates, right, and puts the carboxyl group on the biotin. All that's in that little reaction arrow, and on the right-hand side, we end up with our carboxylated biotin, which is the carboxylated enzyme. And O, being the first product, or products in this case, is the leftover magnesium ADP and the phosphate. Our modified enzyme, represented by the little E slash, moves over to the second active site, right? Or in the case of bacteria with the compartment with the different enzymes, it's the carboxyl transferase component, right? It binds there along with another substrate, P, represented by acetyl-CoA here, and it transfers that carboxyl group from biotin onto acetyl-CoA to make malonyl-CoA. That's what we were just describing on the last slide, how the acetyl-CoA gets a new carbon, a new carboxyl group. That will regenerate the biotin without a carboxyl group, the B, C, CT, and the carrier protein all back to where they were started. R leaves as malonyl-CoA, and I can repeat the whole reaction. And what I want to focus on here is that second half reaction, where we take the carboxylated biotin on the carrier protein, add our second substrate, acetyl-CoA, and how we get to malonyl-CoA. Okay, so that mechanism is shown here. In the very top picture on the left, you can see we have a biotin. You notice the double ring structure of biotin with the thiophane ring and the urido ring. And it has the carboxyl group attached. So this pair of oxygens and a carbon here represents our CO2 that's been attached to biotin at the N1 position. Right? So we have the CO2 group in the presence of the enzyme and the acetyl-CoA over here. It forms this carbonyl, spontaneously decarboxylates in the active site, leaving biotin with an oxyanion here at the top where the urido ring was. At the same time, we have a base in the enzyme, some generic base. We may know what that base is, may not. It deprotonates this methyl group, right, leaving the double bond to migrate between the two carbons, again generating an oxyanion. The enzyme has a pair of oxyanion holes that stabilize this pair of oxyanions to stabilize the intermediate or transition state. After that transition happens, which looks like this at the bottom, right, we have a collapse of both of those carbonyls. Right? The collapse of the biotin carbonyl promotes the deprotonation of that general base. So it's acting as a general acid in this case, regenerating our biotin so it can go get another CO2. And we also have collapse of the carbonyl on the acetyl-CoA, where the methylene group then attacks the carbon of the CO2, generating a new carboxyl group and a carbon-carbon bond, and that's shown on the right. So our biotin's regenerated, minus its carboxyl group this time, and we have an extra carboxyl group attached to our acetyl-CoA. Okay, so just like we said before, taking a acetyl-CoA, deprotonating its methyl group, attacking the carbonyl of bicarbonate, which came from CO2, and putting that CO2 or bicarbonate on acetyl-CoA to make malonyl-CoA. And then of course it can leave and do the reaction again. Okay. What I didn't show between the last slide and this one, it's mentioned in step 3 up here, but I didn't show a picture of it, is this malonyl-CoA we just made is still on CoA, right? So CoA pictured at the bottom here, right? You got the adenosine group over here, a couple of phosphates, another phosphate, the pantothenic acid, and then that's that sulfur, the business end of it, which you learned was made from a cysteine, right? So this SH at the end of it is the business end of CoA, and that's how all our acyl groups are attached. And what we're doing in the step 3 at the top is taking our malonyl-CoA, the malonyl group we just made, plucking it off of the CoA, right here, and putting it on a similar, almost CoA-like thing, right? It's still a pantothenic acid there. But instead of having the other parts of CoA, like the adenosine group, it's a protein where the pantothenic acid's been attached. So it's an acyl carrier protein. Don't confuse this with the biotin carboxyl carrier protein, which was part of our previous enzyme where the biotin was attached. This is a separate protein. So it's a carrier of our newly formed malonyl group. So that's listed up here on the right. We've transferred the malonyl group from malonyl-CoA onto malonyl ACP, or onto ACP to make malonyl ACP. Similarly, I take an additional acetyl-CoA that I've done nothing to and transfer it onto a separate ACP. So now I've taken an acetyl-CoA, transferred the acetyl group onto ACP. I took a second acetyl-CoA, carboxylated it to malonyl-CoA, and transferred the malonyl group onto ACP from CoA. So I have these two moieties up here, an acetyl ACP and a malonyl ACP. This carboxyl group on the end of malonyl is the one I just put on. Now, I know I just put it on, and it's the one that's about to leave. So there's no net addition of carbons here, right? We're just combining carbons from the acetates. The new carbon I just put on with my acetyl-CoA carboxylase is the same one that's going to leave in the next step. So this carboxyl group leaves, right? There's a decarboxylation, and it's a favorable decarboxylation because malonyl-CoA is a beta-keto arrangement, so that's not stable. So it decarboxylates, leaving a carbanion behind, which is an excellent nucleophile, and will attack the carbonyl of the acetyl ACP. So we have this carbon-carbon bond forming, right, at the expense of decarboxylation. Where did the energy for this come from? Well, remember I spent an ATP earlier to make that carboxyphosphate in order to put the CO2 on the acetyl group. Okay? And now I'm letting that go, so I'm expending that energy now to form this aceto-acetyl ACP. Now this might look very similar to ketone body formation, and it is. I've made another aceto-acetyl group, but it's not on CoA this time, it's on this carrier protein, and that makes a difference. What we would normally do here next in ketone body formation to prevent decarboxylation, right, and to not make acetone is to reduce it. So we're going to do the same thing here. We're going to take this group and reduce the blue carbonyl down to a secondary alcohol. Right, so we're going to form a secondary alcohol at the expense of an NADPH. We're reducing this, and it makes the 3-hydroxybutyryl ACP. Okay. Next step, we're going to lose a water molecule, so we're going to dehydrate it. So this hydroxy group and one of these protons is going to leave as water, and we'll form this crotonyl ACP. This is a very stable system because it's conjugated. Okay. Finally, we'll take this double bond and reduce it to a single bond to make the butyryl ACP. And what I've accomplished is, starting with acetyl ACP in blue up here, which had 2 carbons attached, I now have 4 carbon fatty acid attached. Okay. If you think back to when we took our fatty acids apart during degradation, we did a series of reactions, kind of the reverse of this process. So instead of making this, let's imagine this bottom molecule, this butyryl ACP, what would you do to degrade it? Well, we had an acronym for Ohio State. The first one would be to oxidize it, and that would be putting the double bond on here. Then we hydrated it, that's the reverse of this, putting the OH on the beta carbon. Then we oxidized it again to put the carbonyl there. And finally we had CoA come in and attack this carbonyl and lose this acetyl-CoA, which would normally be a CoA instead of ACP here. And our fatty acid would then be 2 carbons shorter. So this is simply the reverse of that process. None of the same enzymes, of course, it's all different enzymes, but the reverse of those steps. So here, instead of doing Ohio State, we're going to do the exact opposite of each of those steps. So the end of that was the state of the sulfur transfer. So we're going to transfer this off of one sulfur onto the carbon. So we're going to lose the ACP. Then reduction, dehydration, reduction, which is the opposite of oxidation, hydration, oxidation. So I finally have a butyryl ACP. What do I do now? Imagine taking this butyryl ACP at the bottom and putting it where the acetyl ACP is at the top of the screen. I take yet another acetyl-CoA, carboxylate it to malonyl-CoA, transfer the malonyl group onto another ACP, and I have this molecule once again. I take my butyryl ACP at the top, another malonyl ACP here, decarboxylate it, add the 2 carbons together, reduction, dehydration, reduction, and now this thing will be 6 carbons long, right? I take that molecule, put it at the top, repeat, repeat, and repeat, and it keeps growing by 2 carbons. Right? Every time I do this, it grows by 2 carbons. So now you see why most fatty acids are going to have an even number of carbons. I start with 2, and I continually add pairs of carbons, it'll stay even. So how do I make an odd chain fatty acid? Well, to make an odd chain fatty acid, I still can only add 2 at a time. So if it's going to end up being odd in the end, I must start with an odd number of carbons. So here, instead of starting with acetyl-ACP, I'll start with a propionyl. Right? So a propionyl-CoA converted onto a propionyl-ACP, and then simply keep adding 2 carbons at a time, and it'll remain odd until I reach my final tally of carbons. Right, and then a little joke at the end. The liver does a lot of this synthesis for us, and the liver does a lot of enzymes, and it's a lot of enzymes, there's a lot of reactions. And it's one of the organs, one of the few organs that we knew of, even the ancient Greeks knew of, that it could regenerate itself. Right? If you cut off a muscle or you cut off a limb, it generally doesn't grow back. A lot of organisms, they do, but in humans, we don't. But we do have one organ that will grow back, even if you cut away 90% of it. As long as you leave the right 10% around the hepatic portal vein and such, the liver will grow back. It won't be perfect, but it will regenerate itself. It's the only organ that can do this, as far as we know. And even the ancient Greeks recognized this. Right? They came up with their myths back then, and one of them was Prometheus. And Prometheus was the myth of the god that brought fire to humans. And Zeus punished him for doing this. It was considered something he shouldn't have done. So he chained him to a rock and had an eagle come along and eat most of his liver every day. Of course, it would regenerate. So this myth evolved or came out of the idea that they knew the liver regenerated.